First, just let me say thanks very much to Edith and Peter O'Donnell for the opportunity to be here, and also to the TAMEST organization for putting on what has been just a fantastic conference and all that you're doing for the state of Texas. As was mentioned, I'm a chemical engineer, and so as an engineer, looking forward, so many of the challenges of the next 20 years in healthcare, in energy, in transportation, really are going to be highly focused on how do we discover entirely new solid and liquid materials with properties that are specifically tailored for technological applications. And so what we do in our group is try to answer the question, how can molecular scale theory and computation really accelerate that process for finding new materials and aid in the understanding of the material behavior in those applications. And so I'll just try to highlight a couple of those in the talk tonight. If you think about this in a really naive sense, if you want to create an entirely new material with novel properties, one way you could go about it is just try to select two or more components and mix them together. And these components may individually exhibit some of those properties you'd like to see in the final material. And if you do that, inevitably what you see is that structural order emerges. And this happens actually automatically. It's not something that you have to turn on. Now, it may not be the structure that you like. It might not be the structure that you envisioned. But ultimately, structural order has to emerge because of the interactions between the components and the material. It can't be turned off. So perfect randomness is never an option in condensed phases. This is a basic rule of statistical mechanics. And actually, for an engineer, this is a beautiful and very useful tool because in the end, when you get to the material, all the properties that you care about, its engineering performance, so how it looks, how it feels, its mechanical integrity, how it's going to respond to external stimuli, or even if or how it might respond to the passage of time, depends on how this structural order emerges, what it looks like, and then your ability to tune it. And so one of the biggest areas in engineering science is basically trying to understand how the structural order emerges and how its properties are going to implicate the engineering performance of materials. And here I'm just going to show you what I think is a beautiful example of this. This one turns out to be from Brian Korgel's group. He's in my department. He actually was the O'Donnell Engineering Award winner from 5 years ago. And what I'm showing you here is a micrograph of essentially ligand-passivated lead selenide nanoparticles that are roughly 7 nanometers in diameter. And these nanoparticles, when the solution or suspension that they're in, solvent gets evaporated, they spontaneously assemble into this structure with long-range order, so a body-centered cubic structure. And this type of self-assembly has actually really gathered a lot of attention in scientific literature because it doesn't really matter that it's lead selenide in some sense. You can build this super lattice from self-assembly with super atoms that are semiconductors or insulators and metals, so many different types of properties. And so the applications and the possibility of tuning material properties seem almost limitless when you see these sorts of self-assembly processes occur from solution. But the problem is the dream has really not been realized in some sense at the nanometer length scale, and part of the reason for that is that when you look at the structures that form in these super lattices, they're body-centered cubic structures, or they're face-centered cubic structures. And at the end of the day, these are essentially nanospheres with very short-range interactions, and so one doesn't have access to the zoo of crystallographic structures that you'd really like to impart on these materials. So how can we get diamond? How can you get simple cubic? How can you really dial in the morphology? So one way you can do that is you can start to ask, in a more systematic way, using computations and statistical mechanics, hey, maybe we can just start tailoring the shape of the particles in order to induce self-assembly. And it turns out that molecular simulations are perfectly suited for answering this question because they connect the details of the interactions of the microscopic components to the final structure that you get. And so this is a technique that's actually being carried out by the world leader in this area, and we're fortunate actually to have her here tomorrow for a talk that Sharon Glotzer and her group. And I'm not going to try to steal her thunder too much here, but I couldn't resist pulling this beautiful image from her science paper, which essentially says, look, you can actually now exhaustively categorize hundreds of shapes and using statistical mechanics map out the structures that they assemble into. And so we're now starting to build the sort of knowledge that allows you to design in assembly by shape at the molecular level. Now, one perspective on this is that, yes, one day we will be able to manufacture any shaped nanoparticle that you like, and maybe we'll even be able to do that inexpensively. But at the present time, it would be great if we could get to these structures, diamond, simple cubic, morphologies of your choice by spheres with just isotropic interactions, so nanoparticles that are very cheap to synthesize, like the particles that I showed in the coral slide. And so this is a problem that we've been working on in our group, and our approach has been to take statistical mechanics and essentially turn it on its head. And we're not the only one who's doing this, but the main idea behind the calculations, these so-called inverse calculations, are that you begin the calculation from the output. You say, I want diamond, and then you use optimization as a tool to walk it backwards and ask, what must you have input to get that structure? And so by doing those types of calculations under various constraints that allow you to input whatever knowledge or physical constraints you have about the system, we began to come up with some very surprising results. So one of them is that it turns out that you don't need special shape or patchiness or bonds to get very open structures. In fact, I'm showing you here a couple of different curves. This red curve here is a curve that assembles into the normal sort of boring, dense FCC lattices. But this blue curve actually stabilizes the ground state for the diamond structure over a very, very wide range of densities. And the only difference is it's a little bit softer repulsion than in the case of the red particle. So there's no bonds, no patchiness, etc. Now what's somewhat interesting about this type of ground state calculation is that if you go and look at phenomenological models for nanoparticle interactions, you can see that you can start accessing all of this space between FCC and diamond just essentially by increasing the geometric ratio of the length of the ligands over the core diameter. Now, of course, these models aren't perfect, but this gives you sort of a roadmap, and now we're going together with Brian Corwell and his group to try to synthesize for the first time structures that are in that large zoo that's in between the highly coordinated FCC lattices and those in the diamond structure. Now, I mentioned these are ground states. It turns out you have to do ground states at the current level of computation when you do inverse design for crystal lattices. But we went ahead and did the forward calculations with this mathematical model for diamond and also for another one for simple cubic. And what you see is that these aren't just blips on the phase diagram. These phases actually occupy a very large region in terms of the density and the virial coefficient, which essentially characterizes the solvent quality that's involved in these systems. And more interestingly, these aren't just thermodynamic results. You can go and actually self-assemble in a very short time on a computer simulation from the fluid diamond and simple cubic phases with very trivially simple potentials. And the reason why you can do that is there's no bonds to get you trapped in the sort of kinetic traps that Jose mentioned a little bit earlier that appear in heteropolymers when they try to collapse. And of course, when you're thinking about how you're going to use computational design and theory to help in the construction of new materials, it doesn't have to be just for crystals. And so I'll just give you an example of a role that this type of design can also play in a system where you have a large degree of disorder. And the technological or engineering problem that we're trying to solve here is a really clear one. All we'd like to be able to do, and this is when I say we, this is a collaboration between myself, Keith Johnston, and Jennifer Maynard. These two invited me to be a part of this a couple of years ago, but the technological question is simple. How can we create a liquid form of therapeutic proteins that's appropriate for single-shot injections for at-home treatment of disease? And so the idea behind this is the following. If you look out in the market today, there's 40 or so therapeutic proteins that have been used for a wide variety of, treating a wide variety of ailments from allergies, autoimmune diseases, to cancer. But there are some major engineering problems and challenges that need to be overcome if these therapeutic proteins are going to become mainstream. And those are really all driven by the patient. And they're related intimately to structure, as I'll mention here in a few seconds. So first, in talking about the constraints, these liquid forms of proteins have to be highly concentrated. If they're not, then you have to have multiple injections, or you have to go to a clinic, and it's going to be an IV that a patient is dealing with. On the other hand, they have to be syringeable. So that means the viscosity has to be below 50 centipoise, or maybe even below 20 centipoise, so that you can push it through a needle. Now, when I show this to my chemical engineering friends, they always look at this and say, hey, you can always make the pipe bigger. But, of course, there are always going to be constraints on patient tolerance. The treatment can't be worse than the ailment. So the third thing that you're trying to balance here, and this really kind of gets to some of the contradictions that Tom Lang was mentioning this morning in his talk from P&G, is that these proteins are going to have to be biologically active, so they can't unfold and aggregate so that the delivery mechanism is ineffective. And so in talking about those contradictions that appear whenever you're trying to solve a design problem in engineering, you realize that, look, hey, concentrated means viscous. And if you think about it from an engineering perspective, this is really just a phenomenological equation, but what it's capturing is the fact that if you've got a crowded solution of sticky molecules, then if you concentrate it, it's going to be harder and harder for that liquid to flow. Okay, and so this equation here relates the shear viscosity to the concentration of the protein solution, and the important parameter is up here in this exponent. It's the intrinsic viscosity, and that number, if these were purely neutral hard spheres, would be 2.5. If they're therapeutic antibodies at 250 mg per mil, that number looks, in a measurement, looks much more like 18 or 15. And so the idea is that the stickiness of these particles is what's causing the viscosity problems. And so we could fix this if we just make therapeutic proteins hard spheres. But hard spheres don't really have much of a therapeutic value, and so the question is, how do you get to that point? And the idea is maybe you can through structuring, or turning the therapeutic protein into a complex fluid. Okay, concentration also means denatured or aggregated, as I'm showing you in this unhappy, irreversibly aggregated egg protein state. The idea of proteins have enough room to unfold. They can fire in lower free energy states that are irreversibly aggregated, and you get on a kinetic pathway that you can never recover from. So how do you avoid that in your formulation of therapeutic proteins? Okay, so maybe you might think that biology has already solved this problem. Maybe evolution has already found ways for cells to be, or for fluids to be concentrated, syringeable, have active proteins. And in fact, I sort of accidentally gave away the answer. If you think about it, cells are really these things. They're highly concentrated, they have active proteins, and they're fluid. But the difference is, or one important difference is that these are highly multi-component systems. And really a therapeutic protein delivery system, you don't want to share that property. It really needs to be carrying the cargo that matters. And so if you think about it, you can still learn something about what stabilizes these, or could potentially stabilize therapeutic proteins from mechanisms from the cell. So I've shown the sort of physicist protein picture here. And in the denatured and native state. And the main idea here is that the native state is a more compact state, and so it can be protected against unfolding if it's in a crowded enough environment like in the cell, as long as what's crowding it is inert. And this crowding feature is really an entropic argument. Turns out if I add these molecular crowders, and these could be polymers or sugars to the solution, they would also drive the aggregation of native proteins for precisely the same reason. So it reduces the overall excluded volume of the system. Okay, well, so this may be what's going on in a cell, but in a therapeutic protein situation, we might have a similar effect happening. So it may be possible at high enough concentrations that the native protein can protect each other from unfolding, so that there could be a self-crowding mechanism. And it turns out this is the kind of mechanism that's not easy to directly check with experiments, but you can address with multiscale modeling. And we published a variety of papers in the past decade where we developed systematically multiscale models for concentrated protein solutions. And what came from that, at least one thing that came from that, is this curve here, which shows, and this is just a representative curve for a protein, it shows the fraction of folded proteins as a function of concentration. And all I want you to recognize here is that if you're at very, very high concentrations, proteins don't want to unfold, there's not enough room. And it's only when you start reducing down into the sort of usual biophysical arena that the sequence and solvent details matter, proteins unfold and they get on these kinetic pathways where they go into irreversible aggregation and never recover. Now on the big picture, if you're just thinking about protein solutions, this graph is really useless. And the reason why is that you just can't get up to this type of concentration. And if you ever could, you couldn't push through a needle. So it's not a technologically relevant situation for a bulk protein solution. But there is a possibility to realize it locally, and this is something that I'm going to now present a conceptual picture that looks a little bit prettier than the actual story turned out. And it was actually observed in Keith Johnston's lab, but I think we didn't understand the reasons for it when I got invited into this collaboration. But here's sort of at least the cartoon that we think underlies what's going on. What they observed is that if you add trehalose, for example, or a small molecule sugar, you can drive self-assembly of proteins to form clusters. And these clusters, their ultimate size is just balanced by the weak electrostatic forces that are present in protein solutions near what's called their isoelectric point. So these end up having finite size. Inside of the cluster, you've got self-crowding. Outside of the cluster, the proteins are protected against folding by the sugars. And so when we came into the scene at this point, we suggested that there are equilibrium arguments that might be able to explain this cluster formation. And if that's true, then we should be able to tune the cluster size, maybe the properties of those clusters, and then tune the viscosity of these protein solutions. One thing that came from theory, not computations on this, and I'm really simplifying it here, is that you can make a prediction just from a clustering model from colloid science that the number of proteins in a cluster shouldn't depend on the concentration of proteins and sugar independently, but it's really just the product of those two. And that has big implications for why these clusters weren't initially seen in protein systems. So here I'm just showing you a calculation from the simple theoretical model. This is protein concentration and sugar concentration, and these dotted lines are isocluster lines on this state diagram. And the point is that in the pharmaceutical industry, with a little bit of sugar, and you go up and up and up in concentration, you only see very tiny clusters. And those have been reported in the literature. And in biophysics, it's sometimes the case that you would like to add a lot of sugar to make sure your dilute system of biological molecules is stable, and you won't see clusters there. You really need to get into this region to start seeing the clusters that I'm mentioning. And if you go in and you create clusters in the laboratory, and this is an example from a therapeutic protein 1B7, what you see is that very high sugar concentrations and high protein concentrations, so here it's 220 mg per mil of 1B7 and similar concentration of trehalose, you get clusters, and you can observe them by cryoprepared SEM or by dynamic light scattering. And it turns out that the solution's still clear because of the size and the optical properties of these proteins, and the viscosity is still low enough that you can push it through a needle. And in fact, I mean, this is a very expensive, or monoclonal antibodies are very expensive to make or to buy, and so we've studied this with Keith Johnston more systematically with polyclonal antibodies, but this is a curve that just is testing this idea that maybe equilibrium ideas underlie this phenomena. And the point is the following. If these are truly equilibrium clusters, and this is not definitive evidence that they are, you should be able to adjust their size just by going up and down in concentration. Wait a while, try it again. And this is just a curve showing that the cluster size for this polyclonal sheep IgG is reversible. We started trying to go for the world record on protein concentrations for a wide variety of proteins. I'm just showing you the 1B7 and the sheep IgG protein here. And what we found was that we could achieve lower viscosities in a 267, 275 mg per mil protein. These are very high concentrations for these types of protein solutions when they had equal concentrations of sugar. So it's sort of counterintuitive. You're adding even more stuff, and you're getting much lower viscosities than you get in the pure protein solutions. The one number, or the two numbers on this table I just want to point out, are the intrinsic viscosities. These are the numbers that are 2.5 if these are pure hard spheres, and they could be 18 if it's a protein solution. So what we've seen is we've been able to reduce those down to 7 by the addition of the sugar molecules. So the structuring that's going on in these systems seems to be consistent with having with effective interactions with clusters being more weakly, being weaker than one would expect based on the monoclonal antibody solutions. One thing you might ask when you're doing all these horrible things to the proteins, making them form clusters at high concentrations, is what happens when you dilute them back down. And so this is just an example of the type of biophysical tests that you do when you dilute these protein samples back down, showing that the CD, or the secondary structure in these systems, is just simply the same as you would expect from a very dilute solution of the same therapeutic proteins. And so the last slide I want to show is just results from an animal study that was done in Jennifer Maynard's group as part of this publication, where we tested injecting into mice the dispersion of clusters at very high concentrations for 1B7, a lower concentration solution in an IV. And the idea is that at least the pharmacokinetic parameters look very similar for those three approaches. So it looks like, at least from the mouse's perspective, in these very preliminary results, that forming protein clusters not only gets you to the low viscosity region, but it also allows a possible delivery mechanism which really isn't protein dependent. And so since then, we've been able to do this with a variety of other proteins in collaboration with a pharmaceutical company. So it looks like a promising technology, but one that needs to be tested on many different fronts, and I'd be happy to discuss those ideas with you here later at the conference. So having said that, the conclusions are, look, structuring is ultimately inevitable in condensed phase systems. So this can't be avoided. But this is not a bad thing. It's actually a beautiful thing for engineers because it means that you can tune the properties of these systems, whether they're soft matter crystals, whether they're new device materials, or whether they're therapeutic proteins. And molecular theory and computational tools will help in the guiding of that tuning over the next 20 years. So I'll just say thanks for your attention, and many thanks to all the various graduate students, postdocs, colleagues, and funding agencies who have contributed to this work over the past 3 or 4 years. Thank you.
Excellent presentation. Enjoyed it very much. In the world of surfactants, they use a lot of lyotropic systems in order to have significant reductions in viscosities. Have you considered the use of some of these surfactants and liquid crystal-like systems in order to further reduce the viscosity of your proteins?
We have considered that, and it's actually, it was motivated by the same reason that you just mentioned, that there's a lot known in other fields about how you can reduce viscosity and enhance the flow of those systems. Great companies have been formed to do precisely that. So we've had some success in scientific studies in that type of approach, but you're ultimately limited when you're injecting into the body on what surfactants and what concentration ranges are accessible to you. And so that's the challenge that you're always going to be fighting here is how much of that science can you borrow from complex fluids with the constraints of physiology. But no, it's an excellent point, and I think surfactants and amphiphilic molecules in general are extraordinarily interesting from that perspective.
There's certainly some biodegradable systems
that's right.
that might be considered there.
No, that's a great point. Thank you.